Recording here. All right. So, let me switch it here. Okay, that should be full screen. Let me know if it's not for you guys. Okay, so what we're going to be doing in this lecture is talking about reproduction. We're going to lead up to the point where we get to, at the end of the lecture, talk a little bit. We're not going to get into it too much, but the development of a baby, from very early embryonic stages all the way to full term when the baby's ready to be born, which will kind of really bring us full circle at this point in the semester. Um, so we have a lot to get through to that point. We're going to start really talking about the production of the cells that make it possible for us to reproduce, what we call our gametes. Those cells are produced in what we call reproductive cell division, a mechanism that produces gametes, fancy word for sex cells, which are the cells needed to form the next generation of sexually reproducing organisms, which humans are an example of. The two gametes are sperm in men, eggs or ova in women. The sperm are produced in the male reproductive organs known as the testes. Eggs are produced in the female reproductive organs called the ovaries. Okay. When a sperm and an egg cell unite, that process is called fertilization. And what takes place is the fusion of the sperm chromosomes and the egg chromosomes, which results in what we call a zygote, which is that single cell organism. We can call it an organism because it's a living cell that then undergoes mitosis, which we learned early in the semester. Mitosis, and this was all the way at the beginning of the semester when we talked about cells, involves prophase, metaphase, anaphase, telophase, where one cell can split and become two cells, and then two cells can eventually become, what we are here as full-fledged adults, capable of going through what's called meiosis. Now, meiosis is just a fancier term for reproductive cell division, where we produce those egg and sperm cells. You'll see here on this slide or in this figure terms like haploid, diploid. We see abbreviations like N and 2N, and we see numbers 23 and 46, which I'll talk about here on this page where I get into meiosis in a little bit more detail. All right, reading through. Somatic cells, which include any cell in the body other than a gamete, contain 23 pairs of chromosomes. Now, a pair is 2, so 23 pairs means 23 times 2 means a total of 46 chromosomes. Okay? One member of each parent is inherited from each parent. What we call our maternal chromosomes came from mom. Our paternal chromosomes came from dad. Okay? The two chromosomes that make up each pair are called homologous chromosomes, or what you'll see them sometimes listed as just homologs. Okay. Sometimes I'll say homologous pairs. Okay. A lot of times they're color coded red for the maternals and blue for the paternals, because red and blue, I guess for whatever reason, have kind of been the denoted colors for males and females. Females red, males purple or blue. Excuse me. They contain similar genes arranged in the same way or almost the same order, meaning that if you look at, chromosome number 1 for the paternal, it's going to contain the same things that it codes for as what the corresponding maternal chromosome codes for. Things like eye color, hair color, visible traits, along with a lot of other invisible or invisible things. Because somatic cells contain 2 sets of chromosomes, we call them diploid cells. A di, always remember, means 2. The abbreviation or symbol 2N that we saw here on the previous page is what is used to denote the number of chromosomes 46 that those cells contain. Okay? So we can call a somatic cell just like any typical cell other than a reproductive cell. So this could be a neuron, could be a muscle cell, could be a bone cell. Okay, it could be a skin cell. It could be just, any of the cells that we've talked about this semester. And unless we're talking about sperm or egg, which are described as haploid. Okay, and are those cells that are produced through meiosis, which I mentioned is fancy way to just describe reproductive cell division occurring in the gonads. Gonads is just a fancy word for the ovaries in females, testes in men that produce those gametes or sex cells, where we take the number of chromosomes and we reduce it by half, which means that we have 23 chromosomes in the egg, 23 in the sperm, and the abbreviation N is short for haploid, or the symbol N is what's represented for cells that have 23 chromosomes. Okay. Meiosis is essentially the same thing as mitosis, but you have to just kind of multiply it by 2, meaning that there's not just one round of divisions, but actually 2 successful stages. The same stages are involved. What we have are prophase, metaphase, anaphase, telophase, except now we have to denote them as prophase 1 or prophase 2, all the way down through telophase 1, telophase 2, because there's meiosis 1, meiosis 2. Okay. And I'll kind of just give you a brief description of each of them here on this next page. This is very brief, as I just said. I could go into much more detail. Kind of just describing the difference between the two. Meiosis 1 is where we go from one cell that begins as diploid and we end with 2 cells that are haploid. Okay? So we have one cell at the beginning of this entire process containing 4 chromosomes. By the time we end meiosis 1, we have 2 haploid cells. Okay? 2 cells that contain 23 chromosomes each. Okay? And during this stage, we separate what we call homologous pairs or homologous chromosomes, which we show an example of some right here. Okay, maybe this is chromosome 1. This is the paternal. Here's the red one. This would be the maternal. So we could say chromosome 1 from the male, chromosome 1 from the female that was inherited. Okay. Notice here they have little letters here, big B's, little b's, big A's, little a's, that kind of stuff. Those are what we call alleles, just different forms of the same trait that's being inherited. It's the same gene that's being inherited. Notice, the B's, regardless whether you're looking at the paternal or the maternal, are on the same spot, kind of like what I said. Essentially, they're identical as far as what they code for, but they're not always identical as what, the allele that they have and what's actually being inherited, because mom and dad aren't identical. And that should be, pretty common sense. The first part of meiosis 1 is called prophase 1, and this is where the chromosomes condense, nuclear envelopes dissolve, kind of like what we talked about with mitosis. Where prophase, we see the nuclear envelope kind of disappearing. We see the appearance of those kind of things, to me, also look like spider legs coming towards the center of the cell to grab onto those chromosomes. Something special takes place during prophase 1, and this is called crossing over. Okay, and this is what's actually being shown up here in the illustration, where these chromosomes set up so closely with one another in these pairs that they can actually exchange segments. Okay, now in this case, they're exchanging up here. Maybe they, next time, or then the other chromosome exchanges down here. Okay, where they actually give in this case, the paternal part of that to the maternal, and the maternal gives part of it back to the paternal, which means now they are not completely identical. Now, back here, before crossing over, this chromosome on this side and this chromatid, I should say, on this side are identical. The B and the B are the same here. Now the B and the B are not the same, which is not so much important here, but eventually, once we end up with 4 cells, we're going to end up with 4 cells that are completely unique to one another, which increases what we call genetic variability, something that I would discuss if this was a biology class. But it's very important to ensure that species are not all the same. That way, they're not susceptible to the same diseases and that kind of stuff, and are unique in their own rights. Stuff I really don't want to get into, because that's more biology than physiology. Okay, after crossing over takes place, metaphase begins. Metaphase, remember, think middle, where the chromosomes are now aligned along the metaphase plate, as they call it. Anaphase, they split apart. And telophase, we start to see the cells completely separate. The nuclear envelope is going to reform around each new nucleus, and we see the two cells completely separating from one another. Now, whenever you see the term homologous chromosomes, really, you're only going to see that in meiosis 1. So that's kind of a dead giveaway, that we're talking about meiosis 1. If we're talking about meiosis 2, you're going to see the term sister chromatids. Okay, sister chromatids are these segments within the entire chromosome. This is a chromosome, okay, and the sister chromatids are separate, are separating, as you can see here in meiosis 2. So prophase 1, the nuclear envelopes dissolve again, spindle fibers form. Metaphase, they are aligned down the middle. Anaphase, they split. Telophase, new nuclear envelopes form, and the cells go through cytokinesis, and 4 new cells are produced. So just to kind of reiterate, we start with a diploid cell. That cell becomes 2 cells, each of which is haploid. By the time we divide those 2, we have 4, and we don't go from haploid to anything else. So they stay haploid in meiosis 2. Okay. I'm going to pull up a quick YouTube clip here to kind of just show it. Super simple, short video here. Just kind of help you keep your head straight on what's taking place. Where did it go? Here's the one I like. Okay, so they're talking about here, this is a pair, so a homologous chromosome or homologous pairs. Back here, before they zoomed in, there was another. Remember, we have 23 of these pairs. So all 23 pairs are going to, buddy up like this. And then, during prophase 1, they can exchange segments, what we call crossing over. Okay, so what he just said, I want to kind of point that out. Said that the chromosomes align randomly. Remember that we have 23 pairs. They're only showing 3 pairs here. Okay, it's not going to be like all the blue paternals are going to be on this side, all the red maternals are going to be on this side. They're just going to align. Some reds will be on this side, some blues will be on this side, and then when we end up going through all those rounds of division, we're just going to end up with just an incredible number of different ways that the cells can divide and split those different chromosomes up, once again, to increase genetic variability. So that was anaphase 1. Here's telophase 1. So now we're seeing prophase 2 taking place. Okay, here's metaphase 2. There's anaphase 2. And there's telophase 2. Okay, so hopefully that kind of made things, a little bit easier to envision as far as what's taking place. for the exam, just make sure you kind of, can keep straight. I'm going to ask you, might give you this, homologous chromosomes attached to spindle fibers on a line on equatorial plate. Which of the following stages is this is taking place in? You'd have to know. Since we're talking about homologous chromosomes, you're going to know definitely talking about meiosis 1. If it says sister chromatids, we're talking about meiosis 2. If we're talking about 1 cell to 2, talking meiosis 1. If we're talking about 2 to 4, we're talking about meiosis 2. Okay. And if you can just keep, your prophase, metaphase, anaphase, telophase stuff separate, which you should know from, back when we talked about mitosis, it shouldn't hopefully be too difficult. Okay, let's now talk about men. We're going to talk about the male reproductive structures and the male reproductive responses. And then we'll move on to you ladies, which are, and I'll just kind of say this, a lot more complicated, but rightfully so, because you've got a lot more to do than us men. Okay, we've already mentioned that the testes are the primary male sex organs where sperm production takes place. We've also talked about the testes previously and their role with the endocrine system secreting hormones like testosterone. And testosterone is a hormone that is very, very involved with the production of these male sperm cells, or spermatozoa for more formal. The production of sperm is called spermatogenesis, which literally has the word sperm in it and ends with genesis, which is just a kind of fancy word for creation. It takes place in a type of highly coiled tubes inside of each testes called seminiferous tubules. Inside of these seminiferous tubules are the actual sperm forming cells called spermatogenic cells. Their name is telling you exactly what they do. Spermatogenesis is performed by spermatogenic cells. There's also within the seminiferous tubules what we call Sertoli, or sustentacular cells, which are supporting cells helping to form what's called a blood testis barrier, which prevents our immune system cells from attacking our own developing sperm cells. And then located between those seminiferous tubules are Leydig, or interstitial cells, which secrete the testosterone that I've mentioned. Now, the testes are enclosed in a sac called the scrotum, a sac consisting of hair and superficial fascia hanging from the base of the penis. There's a vertical scrotal septum that divides the testes into the left and right compartments. It helps create an environment inside where those testes are that is about 3 degrees Celsius below that of our internal body cavity temperature. This is very important because it helps with proper development and storage of the sperm. Sperm, if they are at that 37 degrees Celsius or even warmer, really don't like it and will actually start to die off. Okay? 2 muscles associated with the scrotum help increase, or I shouldn't say pressure, but maintain this temperature. I don't know why I typed in pressure, but maintain this temperature. A dartos muscle, which can contract and cause wrinkles in the scrotum, and that thickens it to reduce heat loss when the external temperatures are cold. And the cremaster muscle, which can lift the entire scrotum up closer to the body when the external temperatures are cold. When the temperatures are hot, those muscles both relax and help the testes stay cooler. I won't really get into those too much more than that. Okay, let's talk more about spermatogenesis. Once again, it's the production of spermatozoa, or sperm, as we kind of more typically call it. Inside of those testes, as I just mentioned, are those seminiferous tubules where the sperm are produced. 2 rounds of meiosis take place where we reduce the chromosomes from 46 to 23, or 23 pairs, or excuse me, at 23 pairs, so just 23. The sperm containing 23 chromosomes are considered mature. Now, a key difference with spermatogenesis that separates it from oogenesis, the production of eggs, is that spermatogenesis doesn't begin until around puberty and will continue throughout a man's life. Oogenesis is kind of crazy. It starts actually before a female's even born, and then it has this arrested period of kind of things are in pause, where everything just is put on hold until a female hits puberty. And then a female only goes through oogenesis until she hits menopause, and then her reproductive life is over. Men, technically, we can produce sperm and get a female pregnant until our dying day, whether that's if you die young or if you're 105 years old, because men can produce sperm throughout their life. Now, men are more fertile, much more fertile in their younger years, and they produce a lot more vital sperm in this process. Now, let's talk about this process. It begins with cells called spermatogonia, a plural form of spermatogonium. Spermatogonia are located near the basement membrane of the seminiferous tubules that squeeze through the blood-testis barrier that I just mentioned. They then go through developmental changes, becoming primary spermatocytes. Now, both spermatogonia and spermatocytes, these primary spermatocytes, are diploid in number, meaning that they are going to be abbreviated here with 2N, or we could say that they are containing 46 chromosomes. Okay? Now, these primary spermatocytes are the cells that begin meiosis 1, and that means, and this takes place once again after puberty begins, they divide into two haploid secondary spermatocytes. Secondary spermatocytes, now we see here, are labeled with the letter N, contain only 23 chromosomes each. Okay, or we can say each chromosome has 2 chromatids. Okay, so after we formed these secondary spermatocytes, we would expect the cells to completely separate through cytokinesis, but we see that the cells are still connected here by way of what's called a cytoplasmic bridge. We see the same thing once we've run through meiosis 2, when those 2 cells become 4 what we call spermatids. Okay. Now, the importance of these cytoplasmic bridges is to account for, at least they believe anyway, the synchronized production of sperm in any given area of the seminiferous tubules. Meaning that these cells are all going to kind of develop at the same time. And so if you look at this just small segment of a seminiferous tubule, that means all the sperm in that area are probably going to be developing at the same time. Now, looking at the picture here, these spermatids are really not a typical shaped sperm. And they're really kind of stubby, and they don't have much of a tail. Okay. The final stage of spermatogenesis is called spermiogenesis, where those haploid spermatids develop into the sperm. No cell division takes place during this process, but they just kind of mature, the tails grow, and the cells are then going to be ready to be released, which is a process called spermiation. So keep your head straight. We've got spermatogenesis. This is the entire process. Spermiogenesis is the development of those immature spermatids into mature spermatozoa or sperm. And then the release of them is called spermiation, where they're released out of those seminiferous tubules and will be carried to a structure that we call the epididymis, where those sperm are stored. And here's what a sperm should look like. It's very, very, detailed and very, very organized. Each day, about 300 million sperm are able to complete that process of spermatogenesis. And just think about that number. 300 million are produced every day once a man hits puberty. Now, that number probably definitely goes down as they get older. But just think about, the fact that only one sperm is needed to make a baby. And what that means is a lot of sperm are produced, but really have no role in doing anything. And so we'll just kind of essentially die off. Structurally, they're about 60 microns in length, contain several structures that help adapt them for reaching and penetrating the secondary oocyte, the egg cell that was ovulated, or we'll talk about ovulation, probably get to that on Wednesday. 2 major parts exist to a sperm. It's head, where the nucleus is, meaning that's where the 23 highly condensed chromosomes are located. Covering the anterior 2/3 of that nucleus is a kind of structure that plows into and burrows into the egg. That's called acrosome. It is full of enzymes that are released to help penetrate the egg. So it kind of helps burrow in there, performs what we call extracellular digestion. Um, Capacitation is a functional change that sperm undergo within the female reproductive tract, allowing them to fertilize a secondary oocyte. We'll talk more about capacitation towards the end of the lecture. Tail is the rest of the sperm. It's got several parts to it. The neck is a constricted region made up of microtubules that comprise the remainder of the tail. The middle piece is very important because this is where you have the powerhouse of the cell, the mitochondria, which gives energy that allows the tail to whip itself. Principal piece is the longest portion, and then it tapers to form the end piece. Okay. So that's kind of the structure of sperm. But sperm are not the only thing that are ejaculated when a man goes through ejaculation. There's also a lot of other fluids that come from the secondary male sex organs. Now, not all of these secondary male sex organs are containing or releasing fluids. Some of them are just going to be carrying or storing the sperm that we just talked about. All right, on the back side or posterior side of each testis is a separate structure called the epididymis. And I would highlight the fact that this is the site of sperm maturation and storage. So this is where those sperm, after being formed in those seminiferous tubules, are carried. In the maturation process, this is where those sperm develop their tail and acquire their motility, which enables them to fertilize the egg or possibly fertilize the egg. It takes about 2 weeks, 14 days for those sperm to mature. Sperm can remain viable here for up to several months, which is why, when a male has a vasectomy where they choose to no longer, try to make a baby, they wait several months before they have a male come, give a sample and make sure that the procedure was actually successful. Any stored sperm that are not ejaculated by that time are eventually phagocytized by cells within the epididymis. Like I said, we produce millions and millions and millions of them a day. More than likely, a lot of them are just going to get destroyed or eaten up in this case by those phagocytes. The epididymis also contains smooth muscles that can contract during ejaculation, which would force the mature sperm into this ductus or vas deferens, which is kind of a cool muscular tube that delivers the sperm into or towards the seminal vesicles. The duct has a very narrow lumen, as well innervated by sympathetic fibers. Like the epididymis, the vas deferens can also store sperm for several months. Now, this is the structure that is snipped or clipped or whatever you want to say when a male has what they call a vasectomy. And you can probably figure out why we call it a vasectomy. A pair of glands that contribute to the secretion known as semen are these seminal vesicles. These are posterior to the base of the urinary bladder. We'll talk about what those substances that they secrete are. They secrete their substances into seminal vesicle ducts, and along with the ductus of the vas deferens, create what's called the ejaculatory ducts. And each ejaculatory duct passes through the prostate gland and empties into the prostatic urethra, which is just the function of it, to force the sperm and secretions, I should say, from the seminal vesicles into the urethra. Prostate is what surrounds the urethra and ejaculatory ducts and is located immediately inferior to the urinary bladder. Secretes a milky, slightly acidic fluid into the urethra that helps with increasing sperm mobility and viability. Mobility means they can swim better, and viability just means that they'll, stay alive. The final glands I want to talk about are the pair of mobile urethral or Cowper's glands, which are found at the inner end of the penis, just below the prostate, and they are what produce a clear alkaline fluid, lubricates the head of the penis, and protects the sperm by neutralizing any residual urine. because the urine is slightly acidic, as we've just recently learned about, and sperm really don't like that acid. They also secrete mucus that lubricates the end of the penis, decreasing the number of sperm that are damaged during ejaculation. This is where that kind of pre-ejaculate fluid comes from, that comes out and trickles out before a male, powerfully releases the actual semen. Okay, the urethra, we talked about in our previous lecture, is a shared reproductive and urinary system structure in men, carrying urine from the bladder during urination, as well as semen during ejaculation. Part of the urethra is located within the penis, which serves to deposit semen into the vagina. During sexual activity, blood vessels in the penis fill with blood, allowing it to become erect and able to enter the female reproductive tract. We'll talk about erections and that kind of stuff here in just a little bit. Let's first talk just a little bit more about semen, which is a mixture of sperm and seminal fluid, typically having a volume of about 2.5 to 5 mils, with 50 to 150 million sperm per milliliter. Meaning, once again, when a man ejaculates, they're releasing a heck of a lot of sperm. Only one of those is required to fertilize an egg. There's just a lot of waste in there. Slight alkaline pH 7.2 to 7.7 due to the secretions from the seminal vesicles. Semen provides transport medium, nutrients, and protection for the sperm. Coagulates or clots after ejaculation due to fibrinogen proteins. Remember, fibrinogen, back when we talked about blood, is what caused our blood to clot. Well, fibrinogen found in semen helps cause it to clot. Liquid portion of semen contains substances that help nourish and activate sperm cells. Normal semen is about 10% sperm and about 90% other stuff, which comes from the prostate, which secretes about a third of the volume of semen into the urethra. The secretions from the prostate contain vitamin C and several other enzymes help activate the sperm. Bulbourethral gland produces mucus, as I said earlier, neutralizes acidic urine in the urethra just prior to ejaculation, has a little volume to semen, and the seminal vesicles secrete several substances, including an alkaline fluid neutralizing acids of the vagina, fructose, which provides energy to the sperm, prostaglandins to stimulate smooth muscle contractions within the female reproductive tract, as well as to facilitate the movement of sperm up the female reproductive tract, and fibrinogen, which I just mentioned, is what helps the semen coagulate or clot after ejaculation. Okay, now we can talk about the male reproductive response and the hormones involved. Okay, now you hear in TV and movies, and it's kind of popular to say that men don't really think with their brains; they think with their reproductive anatomy. Obviously, that's not true, but it does kind of relate to kind of what I want to talk about right now with what we call the brain-testicular axis, which describes the interaction that exists between parts of our brain that we've talked a lot about this semester: the hypothalamus, the anterior pituitary, and the testes. This is the interaction that controls spermatogenesis, which once again is a process of sperm production, or sperm cell production, as it says there by the testes. Once again, begins at puberty, and at puberty, certain hypothalamic neurosecretory cells start to increase their secretion of gonadotropin-releasing hormone. Okay, that GnRH is produced by the hypothalamus that triggers the release of two hormones from the anterior pituitary. We talked about these hormones previously: luteinizing hormone and follicle stimulating hormone, or LH and FSH, both released by the anterior pituitary. Kind of do some of the same things, but also do their own individual things. Luteinizing hormone stimulates the interstitial cells of the testes to secrete testosterone. Okay. Testosterone is synthesized from cholesterol in the testes and is the principal androgen. Utilizing a negative feedback mechanism, testosterone suppression suppresses secretion of LH and GnRH. So meaning that if testosterone levels are really high, we're going to shut down the release of GnRH from the hypothalamus and luteinizing hormone from the anterior pituitary, which means that we'll kind of decrease the amount of testosterone we're producing because we don't want way too much and we don't want to produce more and more and more if we already have enough as it is. Follicle stimulating hormone, or FSH, acts indirectly to promote or stimulate spermatogenesis. FSH and testosterone act synergistically, meaning that they work together on the sustentacular or Sertoli cells found in the testes to stimulate the secretion of ABP, androgen binding protein, which is released into the lumen of those seminiferous tubules and the interstitial fluid that surrounds the spermatogenic cells. Remember, the seminiferous tubules are the tubules inside the testes where the sperm are produced. Spermatogenic cells are the cells that actually do the producing of the sperm. ABP is going to help keep testosterone levels high, which allows for the final steps of spermatogenesis to occur within those seminiferous tubules. Okay. Inhibin is not a hormone of the anterior pituitary, but it's released by the sustentacular cells of the testes, will inhibit FSH secretion by the anterior pituitary. So I've been mentioning testosterone. There's also what we call dihydrotestosterone, or DHT, which is just made when testosterone is converted. Produces several effects. Some of these we talked about before. Some of these are kind of more brand new or a little bit more detailed. Testosterone and DHT promote prenatal development of the male reproductive system structures that we just talked about. The ducts, the descent of the testes, development of the external genitals, and even possibly certain regions in the male brain. Kind of, once again, does kind of reemphasize that men actually kind of do think with their male anatomy, although it's not like you would think. Development of the male sex characteristics, like the enlargement of the male sex organs, the penis gets bigger. Development of masculine secondary sex characteristics, the larynx gets bigger, causing the deeper voice. Bigger muscles, bigger bones, facial hair, underarm hair everywhere, as men are super hairy. Development of sexual function, things like sexual behavior, spermatogenesis, as we've talked about. Male sex drive or libido comes from testosterone and DHT. And then the stimulation of anabolism, causing protein synthesis, which is what causes that heavier muscle and bone mass in men. Figures here on the next page just kind of show you the relationship that exists, trace back to the hypothalamus, triggering the release of, or where we release GnRH, gonadotropin releasing hormone, triggering the release of FSH and LH, and what they do. And this is all stuff that would be taking place inside the testes. The spermatogenic cells, which are the cells that produce the sperm that we talked about, and the ABP that was involved, testosterone and how it's all involved. Testosterone produced by those Leydig interstitial cells in response to LH that we talked about on the previous page. So you can kind of read through this. I think really kind of include it because it's in your textbook. Over here on the right is a negative feedback loop demonstrating what happens if our blood levels of testosterone are too high, which it feeds back, as we talked about, to the hypothalamus and the antipituitary, telling us not to release as much LH, because that means we would be producing more and more and more testosterone. And if we stop the release of those, that means our testosterone levels hopefully can return back to normal. So basically just illustrating what I talked about right back here on the previous page. Okay, now that we have an idea of the hormones involved, we can actually talk about the male sexual response. And there's a lot of information in here. I wanted to do that just to kind of give you an idea of just how complicated, but how important this all is. I'm going to break it down into kind of 3 stages or 3 processes: erection, emission, and ejaculation. And I've got a kind of figure here on the next page to show you 2 of those, at least, erection and ejaculation. Emission's kind of in the middle, and I'll kind of show some stuff about that here as we go through. First thing, let's talk erection. Upon sexual stimulation, penis becomes enlarged and stiff. We call this an erection. It is a parasympathetic reflex coordinated by the spinal cord. During erection, parasympathetic nerves extending from the sacral portion of the spinal cord to the penis release nitric oxide. Nitric oxide we've talked about previously. We learned it is a gaseous hormone. It is a trigger, or it triggers vasodilation. So it causes smooth muscles in the walls of the arterioles that supply the penis to relax or dilate. So there's the vasodilation, allowing large amounts of blood to enter the blood sinuses of the erectile tissue. So allowing spaces within the penis to essentially engorge with blood. As these sinuses expand, veins that drain the penis are compressed, which slows blood outflow. So essentially the blood is kind of stuck inside the penis, not forever, but it's kind of stuck in there for a period of time. And this combination of dilation of those penile arteries or arterioles, expansion of the blood sinuses, and compression of the penile veins is what causes the erection. This filling of blood in the chambers is called vasocongestion. Major stimulus for erection is mechanical stimulation of the penis. So like touching the penis, mechanically stimulating those very sensitive receptors within the penis. These provide direct input to the erection integrating center, which is in the spinal cord. Now, that is what's shown right here in the figure. Signal comes up from the penis, up and into the brain. But you have to keep in mind that direct stimulation of the penis is not the only way to trigger an erection. As it shows here, sights. So like, seeing, an erotic, situation, whatever you're seeing or watching. Smells can even trigger, or just thinking about them. Sounds. Those are all things that can trigger an erection. Okay. This involves descending inputs from the brain, both hypothalamus and our emotional limbic system brain, coming down to the spinal cord. Now, negative stimuli like a bad mood, depression, anxiety can inhibit erection through these descending pathways. Okay. So it's not always a positive, thing with the brain being involved, causing erection. Sometimes it can be the opposite that inhibits that arterial dilation and penile veins being compressed, resulting in the erection. So this side of our figure is the erection. Over here on the left side is showing you all the different parts that are going to be involved with emission and more so ejaculation. Emission is going to refer to the discharge of a small volume of semen prior to ejaculation. When sexual stimulation becomes intense, sympathetic nerve fibers from the upper lumbar portion of the spinal cord stimulate contractions of smooth muscles in the walls of the epididymis, the vas deferens, and the ejaculatory ducts, which is going to start the movement of the sperm being propelled into the urethra. Okay, so looking at our figure here, here's a testy on the backside where those sperm are stored is the epididymis. Here's that vas deferens or ductus deferens. Here's the seminal vesicle, which these are all the accessory reproductive sex glands that are going to start moving the sperm or the seminal fluid towards the urethra. Okay. In addition, sympathetic nerves stimulate the accessory sex glands to release their secretions. So that would be the seminal vesicles, the bulbourethral or Cowper's gland, along with the prostate gland. They're all going to be starting to release their substances together. That's going to form the semen. Okay. So semen is formed as these secretions mix with the sperm. Ejaculation is the powerful release of that semen from the urethra to the exterior involving a sympathetic reflex. We just call this ejaculation reflex. Pretty simple, straightforward there. Then somatic nerves from the lower lumbar and upper sacral portions of the spinal cord stimulate contraction of what's called a bulbospongiosus muscle and other skeletal muscles at the base of the penis, causing semen to be propelled from the urethra to the exterior. Okay, that's what's shown right here in this portion of the figure. Now, this muscle contracts in a pulsatile manner. So it's not a sustained contraction. Instead, it's like it squeezes, relaxes, squeezes, relaxes, squeezes, relaxes. Okay. And repeated contractions of this muscle is what causes the release of semen to be a pulsatile manner, not like a straight, forward flow. It's not just a continuous flow. It comes out and bursts. As part of the ejaculation reflex, the smooth muscle sphincter at the base of the urinary bladder closes, so the internal urethral sphincter contracts and prevents the bladder from emptying any urine. So no pee comes out with the ejaculate. Okay. Because of the location of the ejaculation reflex centers in the lumbar portion of the spinal cord, it cannot be interrupted by inhibitory stimuli from the brain, meaning that once it starts, a man can't stop it, or nothing can be stopped. At this point, it's going to happen no matter what. Okay, that wraps us up, at least for now. We'll get talking more about men here at the kind of end of the lecture. Moving on, we'll talk the female reproductive organs and the female reproductive processes. So we'll start just like we do with men. We started with testes, the primary sex organs. We'll talk about the ovaries here. We'll continue with this because it's just about time to quit on Wednesday. Okay, the primary female sex organs we've talked about before. The ovaries, we learned earlier in the semester, secrete the female sex steroid hormones, estrogens, which is plural for a reason, and progesterone. We're going to talk about the production of the female gametes or ova, what we call oogenesis is that process. Kind of two major regions to an ovary exist: an ovarian cortex, which is a region deep to what's called the tunica albuginea, where you find ovarian follicles, which are fluid-filled bubbles that each egg cell develops in. Within an ovarian follicle, we have what we call oocytes, which is a fancy term for egg cells. Now, there's several different types of oocytes because there's several different stages of development, and the follicles have distinctive numbers of cells that surround each of those developing egg cells. We'll get into all that process and all that stuff next time. Deep to that is the ovarian medulla, which really physiologically I'm not going to get into much because there's not really a whole lot to talk about. Okay, I think we'll stop here and we'll start with this on Wednesday. What we call oogenesis is the formation, development, and maturation of an ovum, beginning in birth, before birth, in females. So we'll get into all that on Wednesday. So I will set you guys free, and we'll see you guys on Wednesday.